in the introduction. I apologize for my title. It's, again, one of these titles that you write down when you don't really know exactly what you are going to talk about. What I decided to do for today was to, because I think Mathieu and I are a bit in the neuroscience part, and maybe not all of you know much neuroscience, I decided to divide, to tell you three stories. One story is going to be about just structure, to try to understand in some way a structure of the nervous system. Then a very short part on relating the structure to function. And now most talks I give are about decision making in groups of animals. Is the one I'm really working on right now. But I will go through the three of them. So I'm quite far from the Henry Markram attitude to modeling, so let me clarify for me what is a model, at least for this talk. Naomi Campbell is a model. Also, this little house is a model of a house. And also we have mathematical models, and what they have in common is that the three of them are idealizations of reality. Naomi Campbell is not reality. And the interesting thing about these very simple models is that they can give you lots of understanding about a system. You don't throw all the details, but you have a hypothesis, and you see how far that hypothesis can take you. And I will use this type of very simple modeling approaches to understand some very basic things about the nervous system. Okay, let's get started with trying to find something about how to understand neuroanatomy. Neuroanatomy is something I used to hate. When I started to learn about biology, I moved from physics to biology. And neuroanatomy was one of the worst parts because it was full of names. And even now, I don't know all the names. But at some point, I realized that maybe there's an opportunity for me to understand neuroanatomy. And it's not a very new idea. It's an idea already stated by Ramon y Cajal a long time ago. And it's the hypothesis that there is economy of wiring in the nervous system. He observed that in many of his preparations, and made the statement that the way things are wired up in the nervous system might correspond to a wiring economy situation. But the nice thing now is that we might have the tools to actually show this into more detail. So for the few next slides, I'm going to make the hypothesis that an objective function for the nervous system is to have a total minimum amount of wire. And then I will obtain for a very simple model the optimal configuration, that is where you should locate the neurons in a system to have minimum wire. And I will compare those optimal positions to the real ones. The only species for which we have the full neuroanatomy is C. elegans. For 10 years, people reconstructed the neurons from the electron microscopy of the animal, and we know where is each of the 279 neurons. Also, the full connectivity of the network is the only full nervous system for which we have this data. And then the problem then reduces to the following. Let's imagine that this is C. elegans, and we will have some sensors and muscles on the surface of this ellipse. And then we have the neurons, these red nodes, and we have the connectivity. I will take as a known the connectivity, also where the sensors and muscles are. That I will take as experimental data. And then I will allow myself to move the neurons along the animal and ask myself where I should locate the neurons such that the total amount of wire, the sum of these lengths, is the minimal, okay, is the minimum possible. And then I will compare to experimental data. And this is the result. Here, we plot the optimal position against the real position, and I put it in the units you may want, 0 to 1, it doesn't matter. And then these are the 279 neurons. If the prediction was perfect, you will find that for the 279 neurons, you will have them in the diagonal. The optimal position would be identical to the real position. It's quite good, the prediction, but it's not perfect. You can see that some deviations, but you don't see neurons here or there. So it's very close to the minimum possible, okay? I will go back to those deviations and I will try to learn about them. But first, before going through that, we wanted to check whether this was something that happened in C. elegans or whether we could observe it in other systems. And then we went through the work of doing the electron microscopy in a small portion of the brain of the Drosophila. This is called the lamina. It's a single cartridge in the lamina, so it's really a small portion in the brain of Drosophila. But we did the full reconstruction. We then knew where each neuron was, all the connectivity between the neurons, and this is just some diagrams of the connectivity. As a basic model, you can think of these neurons as rods, connected rods, something like that, okay? And then if you want to do what I showed before in the case of C. elegans, but in the case of Drosophila, you will model them like before, but you have a problem. Now, if you look at the neurons, some of the cylinders are quite thick, and others are much thinner. So you need to model also the size. And the basic way to do that is not to only have a wearing cost, so you separate two connected cylinders, you increase the cost, but you also have a function that does not allow two of the cylinders to be at the same point in the space, and you put the real distances here, so you put some size into the cylinders. And this is the result. So this is the top view of the cylinder, so they are just circles. And this is the optimal position of all the neurons in this portion of the Drosophila brain. And the real ones are indicated by these bars, so there are some deviations, but they are very close to the optimal ones. So it's not only for C. elegans, which was probably a strange case, maybe, but you can see it in other systems. And it was less expected to see it in Drosophila because it's just a sub-portion of the brain. It's easier to have a sub-portion that is suboptimal, but still it's quite close to optimal. But as I was telling you before, something interesting when you do these kind of things, when things don't work, then you can ask what's happening. And something we didn't understand very well is why we had those deviations. And people had speculated what they could be, and they say, well, it's because it's a male and you need a neuron for the penis and blah, blah, blah. But then we thought maybe what's wrong is the way we were looking at the problem. And the way we were looking at the problem was to say that the system had to be close to optimal. And we know that it can be about approximation sometimes. Systems might tend to the optimum, but there are many reasons why they should not be at the optimum. So instead of comparing the optimum with the real system, we did something else. So that picture might be the incorrect one. But what we next did was, instead of just having an objective function and looking at the configuration that corresponds to the maximum or the minimum of an objective function as the optimal configuration that we compare against experiments, we thought that maybe the way to think about real systems, something closer, could be that suboptimal states are possible. But in general, being fairly close to optimality, one would say that the states with higher objective function have a higher probability of taking place in nature. So in that sense, if you have an objective function like that, instead of saying that you have an optimal configuration that you compare against experiments, is that you have a probability of realization of the system in nature, and it has a probability. But the interesting thing is that if states with higher objective function have a higher probability, which is as close as you can get to optimality theory without being there, then if we look at these functions from above or with contours like that, the hypothesis just means that the probability of finding a configuration of the system is just a function of the objective function. And this f is unknown, okay? We don't know it. And for some things, we don't really need to know it. If we look at this graph, this is the objective function, this is the probability with a given f function. But if, let's say, my objective function has this as a and this as 2a, the probability has b and 2b. And independently of how you choose that f, for example, with a different f, here the numbers are different, this is c, but this is still 2c. So the prediction then is that if we allow ourselves to have suboptimal states in this way, then on average, the deviations in x1 should be twice deviations in x2. With two components, that gives you very poor statistics. But then if you have a system with many components, then you have the prediction that the relative deviations of the components depends only on the objective function, does not depend on this f function that we don't know about. Okay, so what does this mean? It means that for components with a higher impact on the objective function, you should expect the least deviation. And for the case of neurons, that translates into the following. Let's imagine again C. elegans, and for simplicity, a single neuron connected with two connections to a sensor. The optimal position for this neuron would be to be close to the sensor, not to waste any wire. But this is just a deviation, x. Now imagine, and this is just how the cost of this x would be. It goes with 2x, two cables multiplied by x. But you have 4 connections, then you are wasting more wire, you move from the optimal, and the cost will change like this, 4x. So the prediction for C. elegans, just because the cost goes with the number of connections, is that because when you have more connections, you have more impact on cost, then you have smaller deviations when you have more connections. So the prediction is that if you have here the number of connections, the number of wires this neuron is using to connect, and the deviation from the optimal position, you should expect the neurons to be here. And what you observe is something like that. So it works fairly well. Okay. So there is no reason or no big reason to believe that maybe one of the deviating neurons is for A and some other for B, but in general, just relaxing that they are not at the optimal position, then you find that deviations can be explained. And you can also take the reverse approach. I have sort of lied to you, assuming that the cost of the wire was the length of the wire, but there are many reasons known in the cost of transporting things inside neurons, or delays or noise, that actually are functions of the length. Now you can ask yourself, how is the cost of the system? What is the cost that best explains what you see in the real system? And then you might consider that you have length to the 1, so length to the square, to the third, to the fourth. And you could do, in traditional optimization theory, the average error you make comparing what you get with different cost functions, comparing with the data. But you go to 1, 2, 3, and 4, you find a very similar average error. The interesting thing now is that we are not comparing only the minimum or maximum of a function with the data, but we also have now the deviation, so information about the full cost function. And that's much more information. If you do then, here for example, you would compare quadratic with the linear case, and you can see the linear is better, but you could do the full-blown Bayesian estimation of which cost exponent is the one that best explains the data. It's actually something we did not consider before. It was 1/2. And actually there are theories about noise in neurons, or even transport theories that also give that number, and people are studying why because one is going like the square root of the length. Okay, so the first part of the talk has been about just considering a way of understanding neuroanatomy. And it was a very simple hypothesis, it didn't have much in terms of modeling. It was very, very simple. And even that simplicity got us the position of neurons in C. elegans, the position of cylinder-like neurons in a bit of the brain of Drosophila, and even the explanation of the deviations from the optimum. Now I want to move to a different problem, trying to show to you there are many ways of doing this. Relations between structure and function. I have chosen a problem that is particularly simple. It is the problem of motion detection. We know from behavioral studies and electrophysiological studies in insects that a way to understand how animals detect motion could be explained by the Reichardt model. Imagine these are two sensors, A and B. This is a delay, and this is a multiplication. There is a minus sign. So the operations are the following. Imagine you have an object going from A to B. So the signal goes here, it excites A, it gets delayed. By the time the object goes to B, excites B, and because this is delayed, they meet here, they get multiplied, okay? You go in the reverse direction. This is the one that is delayed, this is not delayed, so when they meet, they multiply to 0, okay? In a different system, in the rabbit retina, there's a different model proposed by Barlow and Levick, which is given by an and not gate. So when A is active and not B, then this is active. There is now lots of new progress in trying to find how nervous systems really do motion detection, but I want to show to you how you go about trying to understand how you would do motion detection, and this is the way I thought of it. Imagine you have two sensors, A and B, and you can delay A or B, okay? So you have a 2x2 matrix to have things very simply, and let's imagine that you have first A active and then B, so motion, let's say from A to B. And then what is a motion detector? A motion detector is something that gives you as an output something different to when is the reverse. First active B and then active A. So we are distinguishing it as this is the input here and this is the output. It's distinguishing left to right from right to left motion. If it's nothing happens, it should give us an output 0. If it's all light all the time, it also should give 0. It's something like this. Again, it's no motion, so give 0, 0, 0. These are the tricky ones. You might consider that they are like motion or not. If you don't, then you get models that are nothing like the biology. Okay, so let's see how you should think about, in general, motion detection. So these are the outputs. Let's call them R. And then we can operate the inputs in the way we want. Let's say we have a general nonlinear operation like that, and we can operate all the pairs of the matrices, and you have also triples and quadruples if you want, also the single elements of the matrix. But then you have coefficients in front of them. And basically, a motion detector is something that finds for a given operation like that, the values of the parameters that from these inputs give you these outputs, okay? So you can ask yourself, for that funny operation being a multiplication, what are the values of the parameters that give you the right input to output? And actually it turns out that there are several families of motion detectors, but the simplest, the one with least number of elements, is the one with J equals to 1 and K equals to -1. These and these are the only ones that don't go to 0, which is this here, which is exactly the Reichardt detector. So from the full family of possibilities, when that operation is a multiplication, this is the simplest way you can do it. And this is the way that has been found in insects. But then you can ask yourself, what happens if you have different types of operations. Instead of a multiplication, it can be an OR gate or an AND NOT gate, like in the case of Barlow and Levick. And then you find a family, many families of ways of doing it. But you can learn some things. You can tell experimenters, oh, you found an OR gate in your system. Maybe the way you can think about your motion detector could be like that. But also you can help people saying that the Barlow-Levick cannot be a full, it doesn't actually appear as a full model. It's only a part of a model. It has to be complemented with postsynaptic elements. It could be a minus sign, or it could be another AND NOT gate. Where is it? Here. So this is a model that can help experimenters explore the systems, not to have them fixed in a way of thinking as a multiplication, but it could be something else. And actually, the something else is quite rich. Could be, depending on the nonlinear operation, it could be any of these families. But you can propose for different systems, maybe different implementations. Could be this, something that has an opportunity of being the case in the rabbit retina, and maybe this in a different system. Okay, so you can see in this case that the function of the system translates in particular topologies, connectivities between the elements, and particular operations. And that's all I wanted to show with this part of the talk. But now let me move to decision making in groups, which is what obsesses me these days. And actually, there are many ways of thinking about this, but the way I'm going to talk about it today is how you should be taking decisions in the presence, using the information of decision makers that are around you, that are giving you some information. This is the type of experiments one can do. This is Zebrafish moving in a setup. Actually, if I want to tell you the full story, so the way we did this experiment was, let me start it again. So at the beginning, there were 8 fish here, but previously we had trained 4 of them to go to one of the corners using food, another 4 to go to the other corner. And what you see is that the 8 go together, and they don't split into 2 groups, but they decide to go to one place, and then they decide to go to the other place. So this is the animal we have chosen to do experiments, Zebrafish. But the idea of what I'm going to tell you about in the next few slides is that we are using this system as a way of trying to understand possible general rules of decision making in groups. And although we do the experiments in Zebrafish, we also collaborate with people that work in ants or other fish species, and now we are doing work with humans as well. So, again, as I told you, as a way to make models, I'm proposing that you make a hypothesis, and if you're lucky, you get the right one, and see how far it gets you. One reasonable hypothesis used to model some broad facts about how brains function is that brains need to use very ambiguous sensory data and turn that into an estimation of the world. Even if I look at you with one eye, I'm making up the 3-dimensional structure of this room, okay? And that is an estimation. On my retina is a 2D image, okay? So imagine that, for example, if you are a fish and you see something like that, you want to estimate whether that corresponds to a predator or not. So a hypothesis that has been very fruitful in neurobiology is to assume that the brains, to some extent, what they need to do is to calculate the probability that there is a predator out there given what I see. And that has been used to explain many facts in neuroscience. Multisensory integration might be one of the most fruitful ones. And then what I thought was the following. I thought that maybe a way to understand group behavior was the following. Now we have George, and George needs to, again, as before, tries to estimate whether that corresponds to a predator. He is seeing the plants and maybe a bit of the predator. But the nice thing about George is that he has some friends. And now George needs to estimate whether that is a predator given what he sees and given what the other decision makers are doing. If these guys decide that, okay, let's escape, then George says, well, maybe I should then estimate that that's with high probability a predator, and then I should escape as well. So what we did was to take that idea seriously and then to try to work out what theory you obtain when you make the hypothesis that the brain of social animals are then doing something like this, calculating the probability that there is something out in the world, like the presence of a predator, given the non-social information, what you see, and also given what others are doing. I overdid the Photoshop and opened the mouth, so they all look really worried, but I don't think zebrafish look like that. And then if you take that idea very seriously, then I will show you some of the steps, not the full derivation. Then you can obtain some very simple predictions of what animals should be doing. And what we've been doing in the lab is, because there was nothing about zebrafish in the formulation, we checked it in an experimental setup with zebrafish, but also with ants, other fish species, and with humans. We are doing it right now. But let me explain to you the basic idea of the underlying theory. So let's stick to probably the simplest example. Let's have a decision maker that has to choose between two options, X and Y. And for the sake of simplicity, let's make the two options intrinsically identical. So this is the gray value. Fish like to go to dark places. So let's make these two places of equal gray values. But then when George needs to decide, there are already three fish, already at X or going to X or at Y. And what George needs to do by hypothesis is to estimate the probability that X is a good option given the non-social information, so the gray value of those places, and given the behaviors of other animals, okay? So this is the hypothesis, that animals do that. And then some blah, blah, blah mathematics, basically Bayes' theorem, gives you with some very simple approximations that one can check that you get something out of it. And I will explain to you in a slide afterwards what the different parts of the formula are. But just bear with me that there is some mathematics that can get you from the idea to this simple formulation. But this is just the estimation of X being good. Then you need to take a decision. And from the estimation that we just got, then you need to make a decision of whether you choose X or Y. The super rational thing to do would be if the probability that X is good is greater than the probability that Y is good, then choose X, otherwise choose Y. And if you do that, you will see no relevance of this type of mathematical theories to data. And one of the reasons is that it has been shown for many, many years now that animals don't do the super rational thing, even humans don't do it. They do something that is called probability matching, which is you choose X with a probability that is just proportional to the probability that X is a good option, and this is just a normalization factor. And when you just do this, which is actually very nice from the mathematical point of view because you don't add any parameters, you obtain this formula down here. So let's talk about the formula a bit. So it has several pieces. There are three parameters, A, S, and K. And let's clarify what they are. So again, we are in the simple scenario in which George is choosing between two options, X and Y. A is how reliably the non-social information, so the gray value, tells him that X is a good option. S is how reliably an animal choosing an option X tells him that X is a good option. And K is how reliably an animal choosing Y tells George that X is a bad option. And I know the interpretation of those terms because in the mathematics, actually, they are not parameters. They are functions with some meaning I cannot go through. But let's plot the function to see whether we can understand it. So let's just plot this probability of choosing X against the number of animals that have chosen Y and the number of animals that have chosen X. And if you plot this function, you obtain this. So blue is low values and red is high values. So let's get some intuition. Let's fix the number of animals at Y to some particular value. And this is just a function that is low, 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 and then high. So it means that the more animals that choose X, the best is X. No? So in Spain, we say, where does Vincent go, where people go. So this is the mathematics of that expression. But actually, the function is a bit more complicated than that. You can see some very broad predictions. One is actually that the way you should copy other animals actually depends on the number of animals. It's different at low number of animals compared to a high number of animals. The behavior is different. And also, there are these funny lines that are very strange in the function that we should be looking at in experiments. Okay, so that's the theory. We are basing our group behavior on the very simple idea that other animals are telling you how good places are, and it gives this mathematics, and we're just going to check it. So let's do a very simple experiment. We have, again, George choosing between two groups, X, two regions, X and Y, and then we place different number of animals at X and different number of animals at Y. So let's take this very simple configuration, 3 animals at X, 1 at Y. Again, this is the probability of choosing X. Before it was with colors. Now I plotted in 3D so you can compare better with the data. And I also chose a weird way of plotting it, NY. Instead of going from 0 to 4, it goes from 0 to 4 that way, just so you can see the surface. Okay, so 3 animals at X and 1 at Y. According to the theory, would tell you that the probability of going to X would be something like that. You do the experiment 20 times at the average number. And again, to be able to check whether this surface has an opportunity of explaining the data, you go through many different configurations, and that's what you get. And if you want to more closely look at the comparison between theory and data, let's just look at the 2-dimensional lines here, and that's what you get. So again, probability of choosing X, number of animals at X, and different curves for different animals at Y, okay? So I don't allow myself, of course, to fit any of these curves separately. I just allow myself to fit the surface to the data. And it seems to be working well, and some of the predictions of the model are weird. For example, here, this part of the data and the theory is a bit more weird. So you can have lots of animals at X, let's say 8, and if you have already 3 animals at Y, you should not go more to X than to Y. It's almost probability 0.5. And the reason is that for the theory, these animals, you're not just following the side with more animals. These animals are telling you how good those options are. And if the animals are quite reliable already, 8 animals at X are telling you that it's a good option. But 3 animals at Y is already telling you that Y is a good option. So the probability of going to each of those is 0.5. And that's what happens in that region there. Okay, so that worked fine. We got enthusiastic. And then we asked other groups for data. And there was nothing in the model about zebrafish, and that was one of the nice things, that if it's nothing very particular about zebrafish, then it's a formulation that should work in different animal species. It's just about decision-making in the presence of other decision makers. It doesn't need to be zebrafish. It doesn't need to be actually animals at all. But let's look at this data. So Andrea Perna had a recent paper where he looked at ants wandering around. And the nice thing is that you could understand the data as, in a very simple way, as ants going to X or to Y at every point in time. But we didn't have access, no one has access to pheromones, to the concentration of pheromones, but you can see how many ants were before at X and how many ants were before at Y as a proxy of the pheromone. And again, for the theory, you would get something like that. This is the number of animals at X before George. And the number at Y. This is the theory, this is the experimental data. Again, you see biological straight lines. And as I told you before, I had, from the theory, we knew that the behavior at low number of animals had to be different than a high number of animals. You don't see it very clearly here, so what we did was just to transform these functions in a way. We blew this, we zoomed into these regions here. This is the theory again, and this is the data. So you can see, again, in ants that there is a difference at low number of ants and a high number of ants that the theory predicted. And the third example I'm going to show to you is a different fish species. It's Sticklebacks. In this case, the data is from Ashley Ward, from Australia. And it was very interesting because he had the following setup. He had some real sticklebacks choosing between two options, but he also had, these are the real sticklebacks, but also had fake ones made of plastic. So he could have these two fake ones going to X and one fake one going to Y. And then he had, they recorded for 25 experiments the probability of having different final configurations. So you could have all the animals going to X, the proportion to X being 1. In this case, would mean the two animals going to X. You could have some experiments in which all the animals went to Y, some like this is 50%, 25% going to Y. And in some experiment, you had 1 and 1 and a split, okay? But again, we could ask, they had very nice data because they had different group sizes for real fish, but also they had different number of replicas going to X and to Y. So they had 1, 1, 2, 2, 0, 1, 1, 2, etc. And then you could check the same model in all these different experimental situations. And if you ran the model, you could see that it did fit all the data, the same model. Again, they also had a nice situation in which they had now a plastic predator. And you could also trick them by having a replica fish going to the predator. So you could have, again, as before, groups of size 1, 2, 4, and 8. Then you could have 1, 0, 1, 2, or 3 replica fish going to the right, but you always had the predator there. So when you have no replica fish going to the right, you don't go where the predator is. But when you have the replica fish going to the right, then for quite a few experiments, you go to the side of the predator. And the model in this case works slightly worse. I believe it's because of the experiments, but it also works quite well. Okay, so what we got so far is a very simple hypothesis. The hypothesis that we don't go to a group of 8 because they are 8, but because they are telling us whether the option they've taken is a good one. And if you take that idea seriously, you obtain a particular functional form for the theory that it turns out that just by changing, in this case, the parameter K, you go from zebrafish to sticklebacks and ants, and we also have data for humans now. Okay, so to some extent that was too simple, of course, both at the experimental level and the theoretical level. But the nice thing is that if you start simple, then you can build up a bit better. But one of the stumbling blocks in the study of behavior is that we don't have, of social behavior, is that we don't have systems to track animals. And I'll try to make it as fun as possible, but I'm telling you about this because there was nothing in the market that could do this, and if we wanted to continue this line of research, we had to do it ourselves. So the problem is the following. When you have two animals crossing, here you have two zebrafish, they have a point of interaction, and if you know that this is individual 1 and this is individual 2, it's actually not that easy, neither to the human side, but nor to any existing algorithms, whether this is 1 or 2 or the reverse. And the state-of-the-art methods, if you apply them, you obtain the following. Again, this is the video I showed to you before. So the animals were coming from a particular side of the tank. And if they are well identified by the algorithm, they are in color. But if you go to black, it means that they don't correspond to what a human says. And very quickly, in the first crossing almost, they go to black. So what biologists do now is to have lots of PhD students to undo all this black stuff, and they spend months to do it. One of the problems of having physicists in the lab is that they would never do that, but they can spend 3 years doing the tracking system. So, but to show to you what all the kind of algorithms you can find in the literature will give in the future, I made this slide. I'm going to imagine that the state-of-the-art algorithms, when they improve to almost perfect identification of crossings, but sometimes a little mistake. What happens is that it will take, on average, longer to have a mistake, but at some point they get a random assignment. So all that literature is going this way, and we thought that in order to not to have the situation, what we really needed to do was to identify animals. So if that is George and that is Lucy, then we should know that this is George from the picture and that is Lucy from the pictures, okay? Can you distinguish them? Humans cannot do it. So, except in very, very few cases, you can distinguish George from Lucy, but in general, you cannot. So, I have brave students in the lab, and even if humans cannot do it, they tried. And I'm going to explain to you the details afterwards, but the idea is just simply that you can have a signature, a fingerprint of the image of George and Lucy, and no matter how complex that crossing is, you can look again at fingerprints of those images and then see that this guy is that one and that one is that one, okay? And the validation for that video is this one here. So this kind of, this procedure can make a mistake, but it never propagates. So you get the right assignment. So now videos can be of any length. Animals can disappear from view. I can re-identify animals from one video to the next because I know who is George and who is Lucy. So let me explain very quickly the procedure so you can believe me. So we take from the video a portion of the video in which animals don't cross, the easy part, and because they don't cross, I can say that this animal is George and maybe that one Lucy, okay? And I get the stack of Georges and Lucys, and I have those frames as reference frames. But then I know that they have weird, different postures all the time. What should I do? Okay, what I do is the following. I take the image from George, let's say, and I know that George, when I rotate it, it should still be George. If I translate George, it should still be George. So those are properties of George that I should take into account. And in order to do that in the simplest way, what we did was to take the intensity values on any pair of pixels of George and the distances. And you just plot, for example, the sum of the intensity values of every pair and for every pair, the distance between those two pixels, and you just plot the statistics, you get something like that. You could do it with intensity 1, intensity 2 against D in a 3D plot, but that's computationally much more expensive. You can do it also with the difference, and you get a different map. So for all the reference frames, I get a stack of references for George in this abstract space, and for Lucy and for the rest. And when I have a frame that corresponds to someone I don't know about, then I compare it to all of them, and I say, okay, this corresponds to fish 7. It didn't need to work, but it worked fine. This is if you plot it in colors in the setup as you saw before. So it doesn't matter if those crossings are like hell, because they are re-identified later. So the nice thing is that, again, there was nothing about zebrafish in the way we identified the animals, so it could work well for zebrafish and mice, or in case of zebrafish, as I said before, you can interact, you can interfere with the experiment. You can have little roofs in which animals go inside. Oh, he's here, sorry. So animals that go inside the roof and they come out and then you re-identify them. You can also re-identify animals in different videos. So as a way of showing to you how to extend the type of theories I showed to you, decision makers in groups, one thing that I'm sure is wrong in the theory is that in many species there are leaders and followers. It happens in humans, it happens in fish. And let's look at that problem with these kind of videos. Let's say that this is Lucy and this is George. I will say that George is copying Lucy when George's velocity vector is identical to Lucy, okay? So I just look at the correlation between these two velocity vectors. So when they are the same, but I say that George is copying Lucy. George might take a while to copy Lucy, but Lucy is not copying George. So if I do that analysis in this video, for example, and that's going to be Lucy, and these are Georges, George, John, and Thomas. And the line is going to be blue if that guy is following Lucy, and it's going to be red is the reverse. If you look at in real time, it happens something like that. Most of the time is blue. The correlation function sometimes can go transiently into red. When it bounces there, it can go a bit more into red. But it's most of the time blue. So if you do the analysis, then you find that in one day, you can have these 10 animals, and you see that 5, 3, and 10 are leaders of the group. And I don't see the numbers, but, well, I don't see the numbers. but these are the followers. Then, because we re-identified the animals, we want to check 1 week later whether the same animals, if we put them back into the tank, whether the followers are still followers and the leaders are still leaders. You can see that 5 changes, but 3 and 10 are still leaders, and these are followers as before, and that's 1 week later. And you can see that many of the features are the same. So it happens to be the case that in many animals you have leaders that are consistently leaders and followers that are consistently followers that we did not take into account into the theories. Now we are doing different theories in which leadership-followership relations are very important for the dynamical properties. And again, because now we have for the first time a tracking system that can track animals for any length of time across videos, we have developed a toolbox of social analysis for any kind of animal, but we are thinking more of zebrafish, looking at networks, learning, territories. Okay, so I think with this I'm done. I try to give you some impression of the types of ways we think when we model or we do experiments looking at pure structure, whether some very simple hypothesis like wiring economy can give you a lot or not from where neurons are located. There's much more I could have told you about that, but I've told you very little about the structure-function relations, thinking that Mathieu in his next talk will go more into that, but I gave you at least a very simple case in which you could model different ways of doing motion detection and see which ones could be realized in experiments and how can then that help experimentalists to probe their systems. And I try to give you at least a glimpse of how you could think of decision makers in the presence of other decision makers, and how then you can take the information of other decision makers to make better decisions. And I showed to you then different tools that we are using to make better models. We did not include conflict into those models explicitly. We are doing that right now. In humans, for example, that's also how you get in these groups. You could see that they split it. There are some species in which that cannot happen. It must happen. There is a consensus. In some situations in humans, that's also a must. These theories don't give you that. We are working on consensus type of theories as well. And with that, I just want to thank the people that did the work. Most of what I told you before is work from Alfonso Pérez Escudero. He worked in wiring, in the tracking and in groups. Also, Julian was working something I didn't tell you really about, learning in groups. Animals not only decide in groups, they need to learn in groups. So it's a question, a reasonable question, whether to find out whether the learning is done by some of the animals or by more democratically by all of them. The wiring economy work was done in Drosophila by Marta Rivera, and it's a work in collaboration with Dimitri Choklowski from Janilia Farm. And the Zebrafish experiments, the 2-choice Zebrafish experiments were done by Sarah. And with that, I'm done. Thank you.